Today I'm going to be talking about enrichment strategies, and it's essentially concepts and ideas from the laboratory. And they're very specific to the laboratory, but hopefully they'll be able to translate out to some of the other options that we have and some of the other things we've been discussing for the past couple of days. So I'll try to keep my thoughts and comments specific to just the rodents, but sometimes I may wander off because I had the opportunity to work with so many other animals, and there were lots of things that went and were cross species all the way through. In the federal labs, we were all familiar with the Blue Book and also the Guide for the Care and Use of Laboratory Animals, or the guide, that's ALAC. There are also several other books that were available for those of us who are working in the laboratory. And some of you may or may not know that, and I saw this yesterday in the gray pamphlet, the Blue Book gray pamphlet, and it's talking about exemptions from USDA visits to federal laboratories, not federally funded laboratories, federal laboratories. The reason that the folks at the places like the National Institute of Health or the CDC do not get USDA visits is the government can inspect its own. However, those federal laboratories are typically ALAC accredited, and that's a voluntary visit by the ALAC folks every 3 years. There are some differences between the animal welfare regulations and the guide, but they're minor. What I always used to tell folks was that they should be ready at any time for a site visit if you're looking to work with the USDA, and you should be able to meet or exceed both requirements in your laboratory, regardless of whether you're inspected by the USDA or site visited by ALAC. And I put down here requirements versus recommendations. You've got regulations in the Blue Book, and you've got recommendations in the Guide for Care and Use of Laboratory Animals. Some of you may have heard about NCIs yesterday. That's a non-compliance issue, USDA. For ALAC site visitors, you might get an SFI, which is a suggestion for improvement. So the visitations by ALAC are voluntary. You invite the ALAC folks into your facility. One of the interesting things about it is if you are a contract research lab, not a federal lab, you're going to be looking to get and receive federal funding. But if you are not ALAC accredited, you will not be able to receive those federal funding. So it's in your best interest to get your lab up to speed and meet or exceed suggestions from ALAC regarding the Guide to Care of Use of Laboratory Animals in your facility. Over on the right-hand side, these are three things. The guide changes every few years, and that changed from the previous version. The major things to look for working with an environment for laboratory animals was activity, social behavior, and procedural habituation and training. We won't be talking about training today. That's primarily for the non-human primates section. Requirements versus recommendations. And as I said, there are sections in the animal welfare regulations that talk about plans for dogs and plans for non-human primates. And there are no specific requirements for mice or rats, and I'll talk about birds in a second, under the animal welfare regulations. In the guide, it covers all animals, and my veterinary colleagues will be very familiar with the ACLAM Blue Books. ACLAM is American College of Laboratory Animal Medicine, and those are the books that all of my colleagues use to study before they sit for their board exams. So they're very familiar with all sorts of documentation. And I've had an opportunity to help out with some of that documentation over my career. So what are some of the reasons to provide an adequate environment? And we are specifically talking again about the laboratory environment, but it does translate out. You're looking to increase normal behavior and decrease abnormal behavior. Now, I haven't gone into any great discussion about what is abnormal behavior, But typically you'll see all sorts of things that folks don't like. And I will not really discuss too much of that because I've got other things to discuss rather than going into all the potential abnormal behavior issues you might see. That could be a whole nother presentation just on abnormal behavior in laboratory animals. We're going to talk specifically about rodents today and rodent behavior. And in the laboratory, there are significant strain and species differences. I can't even guess right now how many different strains of mice there are. I'd have to get the book on mouse strains. And one of the things to do is and I was looking for a quote that I had from Catherine Bain and Hannah Wurbel about providing environmental enrichment for animals is not a one-shot deal. It's not one size fits all. And that's one of the things that's probably most specific and probably the biggest thing I want to take home today is that each animal brings to the table different things, different behaviors. And so getting your enrichment specific to the animal is what you're looking for. How I would do that, and I didn't work with several of these animals until after I started after school, essentially, but I would initially start off my work looking at the natural history of the animal. How did they live in the wild? And also, what are some of the species-specific behavior patterns that you see in some of these animals? For example, down at the bottom, behavior needs of specific animals. If you're going to keep captive chinchillas, you'll need a dust bath because they need that. And they will use a dust bath in captivity if it's provided. If not, they may not be very happy chinchillas. This is just a short list of the behavioral repertoire of rodents. And you can see for yourself, gnawing, eating, searching for food, burrowing, hiding, defense. I had pulled up a document on rat behavior, and that individual listed over 40 or 45 different behaviors that the rats could do. Quantifying and qualifying those behaviors turns out to be something we call an ethogram, listing all the possible behaviors that the animal can show. And then you can collect some data on those things. It all depends on what you're looking for and what you're trying to record and what specifically some of those animals might be used for. At both of the federal institutions that I worked at, there was a whole list of different possibilities for animals that would be used for. And in one facility, they had upwards of 10,000 mice, and I couldn't even begin to list the number of different studies that those animals were on. However, my bottom line with enrichment and enrichment-specific ideas, or the new term now is behavior management, was these are still mice, regardless of what you've been doing to them and what you want to use them for. So let's start with the basics. It's a mouse, it's a rat, it's a guinea pig, or other animals. Types of enrichment. In the laboratory environment, there are specific caging types for almost every single animal. This is an enhanced cage for rats. Typically, they're called shoebox cages, and some of the older ones may have wire bottoms versus solid bottoms. And I'll ask you to note that in each of these pictures, you might see a different kind of substrate or a different kind of bedding material. That's based primarily on the preferences of either the attending veterinarian or the facility, and specifically requires certain types of bedding. There are some types of bedding that you want to stay away from when you're working with certain types of animals, specifically some pine shavings. But what you'll see here is you'll see several different, and I'll try to remember to point out some of the differences. This is just shavings, I believe, just wood shavings, but I don't know what kind of wood shavings. But the other thing here to see, this is the structural type of enrichment where we've got a platform and several rats in there. And the thing that's a challenge here for most facilities is when you design caging, you have to know what you've got for housing systems and facilities. They're not all standardized. The challenge with that is if you want to build a brand new cage that's great for everybody, those cages may not fit in any of the racks that the research lab has got. And so that does cause a lot of back and forth with vendors and scientists about, well, we want to provide these great environments, but we don't have the money to spend to retrofit our whole entire facility. There are ways around some of these issues. I'll talk about some of them when I get there. Bedding, of course, is one of the easiest ways to enrich the animals. And like I said, there are going to be several different types of bedding. All depends on, sorry about that. It all depends on what they're doing, what they want, like I said, and what's in the facility. Other structures that can be included into some of the cages include such things as tubes, nest boxes, manipulanda, something to move around, or gnawing objects. Again, a lot of this depends on what research is being done, what can or cannot be provided to the animals. And so where I would start would be looking and speaking with the principal investigators to find out what limitations I had to address, or if there weren't any limitations, how far we could go with providing enrichment for some of the animals. I'm going to be discussing social enrichment in the next presentation, so I'll just briefly talk about it here. Again, these are several rats in a cage. I think they're males, but that's my best guess. Rats do well like that, and we'll talk some more about that in the next presentation. One of the ways that folks started worrying about these things was there was a publication in 1985. That was one of the first copies of the Blue Books that I had to look at. That's the USDA regs, with a revision in 1991. And before 1991, there wasn't much about enrichment programs for non-human primates, or a little on exercise for dogs. And there wasn't much about enrichment for some of the other animals. And so what that offered everybody was great opportunities for students doing either theses or doctoral presentations to go ahead and test everything under the sun. And for the most part, they did. Preference testing. What do the animals prefer? The interesting thing about preference testing, up here we've got a cardboard shack versus a plastic shack. The plastic shack is specifically designed to sit in the corner of a shoebox cage. The cardboard shack sits in the center of the cage, and I'll talk a little bit more about that in a second. And the issue with animal preference is the animal preference tests are designed by the scientists to determine what the animals prefer, as opposed to the animals tell you what they prefer, and then you design caging around that. Down at the bottom here on the right-hand side is one of the more interesting experiments that was done looking at cage preferences by the Van Loo group over in the Netherlands. What's really nice about this, and I don't have a picture of it, was they had 4 different cages that the animals could go into, and the little black boxes around the front there, those are photo cells, so they could record when the animals would go in and out of any one of those 4 cubicles, how long they spent in there, and so they were able to quantify their preferences. The other thing that was really interesting about this experiment was you would be concerned if you laid this system down on the floor in a colony room that there would be site preference or preference for one direction versus another. All of these systems were set up on slowly turning turntables, so they never got a chance to have a single preference. In other words, they didn't always go to the top versus the bottom versus the left. They were always turning, so they were still looking specifically at, well, where did the mice spend most of their time, and what substrate was in there that did that. There were hundreds of experiments that were done at that time. I just wanted to talk about a couple of them, and we'll go through a few of them before we're through about that. So again, this is animal preference, trying to figure out what the animals liked. What are we going to put in these cages? One of the things that happened to me on a regular basis was when we would suggest some of these alternative housing systems, there was several folks who would provide lots of pushback on, we've never done that before, you know, and why do we have to change this now? And, you know, is this going to interfere with my experiment? Whenever I would approach PIs, or anyone else in the laboratory. My goal was not to interfere with any of the research. My goal was to enhance the well-being of the animals and then hopefully provide them with much better data, because the animals were much more welcome in their environment and they were much more habituated to living in those environments. Here's a reality check. Over here on the left is a micro isolator, and Dr. McKay talked about bringing in animals. In the back over there is an autoclave. All new, this is mice in particular here, new animals coming into that research facility would go into quarantine, and they would go into these micro isolators. The micro isolators were set up as a micro environment, and everything going in there except for the animals was sterilized or autoclaved before it went in there. So the food was autoclaved, the bedding was autoclaved, and as you can see, you've got these great connectors to the cage so that the individuals working with the animals can move the animals through. But all the animals coming through into quarantine would be put into some kind of isolation system. And this system was in a different campus than the main campus. So you were even more so isolating the animals, and they all went through quarantine periods before they were released into the general population or used in other research facilities. For the vendors, folks supplying some of these animals, you may or may not have received site visits from some of the government facility people. And what they were looking at when they came to visit your facility was how well you were doing with your breeding facility, how you were housing your animals. Were you addressing certain questions that were brought forth in the guide or the Blue Book? Because they didn't want to have to spend all their time rederiving all sorts of clean animals if you sent them some of the dirtier animals. But we still did all sorts of testing on any single animal that came in any of our buildings, and we wanted to provide the best environment for them while they were still in quarantine before they went to either conventional housing or ventilated racks. Over here on the right is old conventional housing, and if you can look closely, they got water bottles hanging from the top, and these are filters on top of these cages. So they're all basically sharing all the air in here, but it's getting filtered onto each cage. This is very, very old caging. The other thing to notice about this is our technician is not wearing a lab coat, not wearing any gloves, not wearing a face mask, not wearing a hair protection. All these things would probably be required or requested, even in static caging like this. A lot of the caging now, and I'll have a picture here in a second of the ventilated racks where each individual cage has its own airflow, and it's segregated by that airflow, and they're all treated individually when you pull them off the rack, even if they're all from the same group. And if there's any of my veterinary colleagues in the audience who know where this picture was taken, this is most likely a visit to a production facility a long time ago, but you can see what they're doing is they're moving animals, and I'm going to assume at the moment they're mice because I can't see any mice, but they're moving them from cage to cage and doing all sorts of things with them, but they're completely isolated from everybody else in the room. Again, we can tell this is a few years ago because, again, the guys aren't wearing much other safety gear. OSHA might not like that ladder. There's all sorts of other issues here, but I wanted to show you this. I do not know where this picture was taken, but I've used it several times to demonstrate, well, this is one way they used to do a lot of this work. There are still isolators in use. Back to the rodent shelters again, and you have to give the vendors in the community kudos because they were trying to do the best they possibly could. Some of the vendors knew something about the animals they were trying to provide enrichment for, and some of them didn't. I had some of the best conversations in my career with people who knew nothing about the animals that they were suggesting were going to use some of this enrichment and telling me that it was the best thing since sliced bread. And I would tell them, I said, well, the animals really aren't going to use that because that's not part of their species typical behavior. However, you have to start someplace. The interesting part about this top shelter here is that it has two exits, essentially, one up top and one out front. The plastic balls on the bottom could either be put in the cages as plastic balls or as split plastic balls. And the interesting thing about this was the rodents would build nests with nesting material inside of these things. And the biggest challenge that we had was the care staff were required to look at these animals every single day, regardless of status, whether they were breeding or not. And the hard part was they said, well, you know, they build these nests, you know, and I can't see them when I look at the cage. This was a static rack. So I had to come up with other ways. I said, well, just pull the static cage out and lift it above your head and look underneath where the mice are, because they won't have all the nesting material on the bottom of the cage. It was a little bit of changing in human factors just to tell people that you may have to do a little different work, but it wasn't too far out of the realm of their system. But they weren't used to doing stuff like that. They were used to running in the room, looking at all the cages, looking at the mice, and going back out and going to the next job. And so I was always cognizant of the fact that this takes extra work sometimes when you put some of these things into cages. The other thing that happens with some of these cages, and it's not every animal, but some of the animals will chew on this plastic. That caused some concern to some of my veterinary colleagues. And you have to address some of those questions sometimes, because the last thing we want to do is make life worse for everybody, whether or not the animal's ingesting the appropriate stuff or not. In the next slide, I believe, yep, this is a picture inside of another micro isolator. And what you're seeing here, I'll show you the different bedding material, and I don't remember exactly what it is, but it's a paper type product. The folks put a running wheel in there. I never really promoted running wheels in cages. I understand why people do it. The issue sometimes is, are you doing something from an enrichment standpoint to make the animal feel better and well-being of the animal, or are you putting something in there to make you feel better? Over on the left is where the airflow comes into this cage. This was actually a dormouse research project. And when I went to the literature early on about this, dormice like to live in trees. And so I recommended, you know, some housing in a facility with trees. Well, this was an infectious disease laboratory, and trees were not part of the process. But at least we had a nice conversation with the PI about this and what they could or couldn't do to provide certain types of enrichment for these animals. They came out of a very enriched environment, and they were into the research laboratory for a very specific reason. So that's one of the things that has to be done when you recommend certain types of things. You have to know the limitations. You have to understand what can and cannot be done, and try to working with both your PI and your veterinary folks to try and see what you can do that will help out everywhere. This is the picture of that cardboard shelter again. And one of the more interesting things about this, and the yellow tab is to keep that in the ventilated rack. So this is a stack rack cage. I don't have any pictures of a stack rack. This houses 6 cages high by 10 cages wide. And basically you're stacking the animals, but they're also individually housed. They've got their own food. They got their own water. Like I said, at the back of this cage is the airflow. What I learned about this was the attending veterinarian in this process told me that he really appreciated the fact that these rodent shelters were here. Yes, it is harder to see the animals. It was much more difficult, but he told me that he got many fewer calls for fighting in mice after they put all these things into the cages. And sadly, a lot of these are anecdotes about things are good. Sometimes folks don't think it's important enough to publish some of that. And sometimes you're not actually testing hypotheses, and you don't actually have something to test or write up about. And that's the sad part. So it's a lot of word of mouth, a lot of anecdotes between facilities. We use this, we use that, we saw this, we saw that. But I tried to go to the primary literature every single time I could to see if someone had tested some of these things versus just tossed them in the cage because they had a really good relationship with their close vendor. these are just some more examples. Again, if you see, this is kind of wooden chip bedding on the floor, but it's also got the crinkle paper. And again, with the crinkle paper, a lot of folks were reluctant to use the crinkle paper because, again, they couldn't see the animals, but the animals love this crinkle paper. It was really easy to distribute and hand out to the animals. And again, if you're dealing with this, you're going to have to look at these animals on a regular basis. And so you have to make some adjustments to do some of these things. You may have to go in and lift up the paper. You may have to go poking around, but the animals really seem to enjoy that stuff. The sawdust doesn't lend itself well to nest building, or at least this wood stuff doesn't lend itself well. These were a couple guinea pigs. The most difficult thing I had to do here was to convince the folks that I could put this pipe in here. Early on, folks learned that you can't use PVC pipe primarily in most of these facilities because everything has to go through the cage wash, all the materials from or be tossed out. This is CPVC pipe, chlorinated polyvinyl chloride pipe. It has the ability to go through cage wash without warping. The other difficult part about this one was I had to buy a Sawzall to cut these 6-foot pieces into short pieces so they would fit inside this is a solid bottom cage and it's about 6.5 inches tall. But the guinea pigs liked it. That bedding down there, I believe it's called Carefresh. The challenge with some of that bedding is it mats easily and doesn't provide the animals with anything other than a softer floor. All depends on the animal, whether you needed to do something like that or not. The other difficult part about providing this was I had to soften all the edges here with some sandpaper so there weren't any rough or sharp edges for these animals. But obviously these animals seem to like that. Again, the most difficult part of this process was the resistance of the care staff who had never done that before. These are cotton rats, and I'll talk a little bit more about those in the next session. So as you'll see later, this one was good because the cotton rats aggression went down once they were provided. These are cardboard tubes. And the interesting thing about cardboard tubes were early on, we were requesting everybody seems to have some kind of cardboard tube at their house, and we were bringing them into the lab. That caused some concerns for our laboratory people because they were afraid of the composition of the glues. We ultimately worked with our vendors, and they now, for use in laboratory medicine, the cardboard tubes are put together with vegetable-based glues, and the animals can do anything they want to them. The interesting thing about the cardboard tubes is we had a colony of rats, and I provided cardboard tubes for them, and 1/3 of them used the cardboard tube as is, 1/3 cut them into small little pieces to use as bedding, and the other 1/3 didn't really care. So that's the law of thirds regardless. So people say, well, they don't use it. Well, I'd rather give it out than not. Again, this cage has got, I believe this might be Sanicel. So it's another cage with some different kind of background. Structural elements. I put these PVC pipes, again, this is CPVC pipes in with the rats, and I had a picture where I had 3 rats and their heads were all stuck on each end of the pipe. And so they were essentially, keeping their eyes covered while they were snoozing in there. But if the rat wanted to use it as a structural element to get closer to the food, that's just fine by me. Same thing with this Kong toy over on the right. And that is, they're using it to get closer to the food, make it a little bit easier for them. That they're not really playing with the Kong toy. But again, did it make the people who put the Kong toy in happier? Sometimes, until they had to clean them and take them out for cage wash. So that was, again, one of those interesting things. So these are rabbit cages. These are standard size rabbit cages. And the difference between some of the cages is this is 5 square feet. That's good for an adult rabbit in the laboratory. These cages do have shelving. And we tried to put some animals together. We drilled holes in some of the plexiglass panels. The panels come apart in the center and you can pull them apart, but 1 rabbit, 2 rabbit, and there's not a lot of room for the 2 rabbits to go. Probably the most important aspect of working with the rabbits like that is someone needs to watch these animals when you try to do some socializations. I'll talk some more about that in more detail in the next talk. But again, how many rabbits? 3 rabbits in a rack. How many racks can you get in a room? You're looking at square footage, etc. So the thing about it is you can have, you can have alternative strategies. And we did several of those when we were working with these animals. Nesting material. Nesting material was an easy one, although originally folks were a little concerned about that. One of my colleagues, one of my veterinary colleagues actually did a cost-benefit analysis because folks said, well, it's going to cost too much to put nestlets in every cage. They actually ran the numbers and said there's really no difference. It wasn't going to cost that much more. And besides, the animals like to make nests. As I said, a lot of the research that went on were certain things like, let's look at what's done here, you know, and the use of nestlets was preferred over the half tunnel. That's one of those cages, or the plastic tunnels, over a marble. Again, putting things in there. Some of the concerns were eye irritation in nude mice or entanglement of limbs. Well, when you look at the data carefully and you look at it, you know, if you're thinking that you've got, you know, 60% of the animals having eye irritation, or, you know, 60 to 80% entanglement of limbs, it works out to less than 1% of some of these animals. And so you have to have perspective. You've got a lot of animals, you got a lot of these things. You will potentially see some of these things with enrichment devices, but usually it's a very, very small, minor percentage. And if you find that, you should address the issue and see what happened, if you know. Again, these mice, they're in an isolator. You can't really see that, but it's a plastic film in front of them. So they've got nesting material in a tube. Again, some of the research that was done, this was Helen Vanderweerd. Nesting material was such as tissue was highly preferred by mice, and there were no major behavioral or physiological effects on the animals. That was one of the keys. A lot of folks were very concerned that there were behavioral issues that would change the animals, or particularly physiological effects on the animals. So by testing that theory empirically, you could provide the evidence and the data that would suggest that providing this information or providing these enrichment types of devices had no effect on the animal whatsoever, but maybe it made a better environment for the animal to do well. Another by Van Loo's group, et cetera. And again, specific to mice, the moderated aggression in male BALB-C mice with olfactory cues, they found that aggression peaked after disturbances such as cage cleaning. Transfer of bedding with urine and feces intensified aggression, and the transfer of nesting material reduced aggression significantly. So they learned that from doing all that work. And it was one of those things that learned an awful lot. Rabbits, crepuscular, nocturnal, they will develop dominance hierarchies. There are some social housing challenges. The research use, these animals were primarily used for feeding mosquitoes and ticks, but also some biofilm. Unfortunately, you can't see the 4 rabbits in that one hut, because I would have had to have jumped in the cage with them, and they didn't like that. So they were just hiding back there. This is that data I was telling you about a little bit earlier. And there was a significant decline in aggression in cotton rats through the use of enrichment. All they did was they put that tube in with these rats. And again, it was the number of cases reported to the attending veterinarian, and it went down over time. So they found that it was a very useful piece of enrichment to provide for the animals. And then I got this from the 7 deadliest words of in laboratory animal medicine from Ron Banks. We have never done that here before. I heard that quite a bit, and then I would turn around and say, well, let's try some of these things, because just because we've never done it before doesn't mean it can't be done. So some people did call me the crazy doctor, but that's okay. I didn't mind that. I was trying to push forward with lots of new things that people had never seen before, and trying to improve the environment of laboratory animals. And we had to work on lots of those things. I have lots of people to thank. I've worked with all these people over the last few years, and I'll leave that up for a second. Several of the techs I had became veterinarians, which was good. I worked with several folks over the years. None of this could have been possible. And I'm sorry, if you're in the audience and I left your name off, I'll have to stick it back on at some point, because I know there's at least one person. So None of this was done in a vacuum. I had lots of help, lots of assistance. So I want to thank all these people for all the assistance they gave me. None of this could have been done by myself. No way. And I think that's my last slide. So now I have time for questions.
All right. Thank you, Dr. Weed. We do have one question in the Q&A that I can see. So if you guys have any additional questions, please put it in at the bottom of the screen in the Q&A section. Dave?
Okay. All right, Jim, got a question here. What types of enrichment, in your opinion, should be considered standard of care for these small mammals? Should there be a difference whether in a research or breeding setting?
Well, as I said earlier, one size does not fit all. And so I would customize to the particular animal and the particular facility. I wouldn't be out telling everybody, well, you got to do this for X, Y, and Z, because then there might be someone who's found something else that works much more effectively, and it's much easier for them to do it, particularly in a breeding facility versus a research laboratory where it's not so much in a breeding facility.
Okay, thank you. Next question. What advice would you give a student or postdoc looking at doing work in behavior?
Ooh, that's a good question. I'd go for it. Do it. If I don't know where you are in your career, but find someone who's doing some of that work, and go with them and stick with it, because there's plenty of work to do. There's no lack of work to do in this area. It's just a matter of you pick an animal and pick something you want to study, and good luck.
Okay, thank you. Next question. What do you think about using dust baths for gerbils?
I was trying to remember my gerbil history, and I think gerbils might use a dust bath. I'd have to double check the literature. I've recently moved and I don't have access to my full library, so I couldn't check everything, sadly. But if I recall, I wouldn't have a problem with dust baths with gerbils.
Okay, thank you. What do you think about this other deadly phrase in laboratory settings? We have always done it this or that way.
I have heard that in my career. We've always done it this way. And my response typically was, well, maybe that's not the best way, given some of the newer information that we have that's out there. In other words, literature. I always tried to back up anything I said to anyone with some documentation, some literature, some research or something, rather than just telling them, well, you got to change the way you're doing things because we don't like the way you're doing it.
Okay. Let me look here in the chat. Okay, so as an expert in research where these animals are, what are some of the top points for pet practitioners, breeders, and owners?
I think the answer to that question, I don't mean to jump away from it, but I believe if you're running a facility, you need to address species-specific behavior if at all possible. Look at the natural history. I understand that you can do certain things with the breeding laboratory or the breeding facilities. I got a picture in the next presentation that discusses this in a little more detail, but it's not a one-size-fits-all. I don't think anyone should mandate that everything should be done for all these animals, but I would recommend more than just housing them to produce more animals with a minimalistic approach. I would, if I were a site visitor to some of these places, I would recommend several things. Not that they would have to be done, but I would recommend some of those things.
Okay, thank you. I think that's we're through with questions.